So our next speakers are Liz Warren and Marc Giulianotti. Liz is the Senior Program Director at ISS National Lab. She's a Senior Program Director for ISS, U.S. National Lab. She's been involved in spaceflight research for over 25 years. She was born and raised in the San Francisco area and attended UC Davis for both her graduate and undergraduate degrees. Her doctoral degree in molecular, cellular, and integrative physiology. For her doctoral work, she investigated the effects of gravity as a continuum on energy balance in rats. She completed her postdoctoral work in cancer biology at the San Francisco VA Laboratory of Cell Growth and in the Neuroscience Laboratory at NASA's Johnson Space Center. She's performed a variety of roles at NASA, including serving as Deputy Project Scientist for the NASA Bed Rest and Artificial Gravity projects. She also spent several years as operations lead and mission control for the ISS Medical Project. She's a passionate science communicator, which I'm sure we'll find out in a minute, and an advocate for human spaceflight and STEM. Marc is a Senior Associate Program Scientist with the ISS National Lab. Part of his activities include managing the Chips in Space program, co-sponsored by the National Center for Advancing Translational Sciences and National Institute of Biomedical Imaging and Bioengineering, as well as tissue engineering and mechanobiology and space programs co-sponsored by the NSF. Prior to joining the National Lab, Marc spent 20+ years working on early drug discovery efforts at the Torrey Pines Institute for Molecular Studies. He received his BS in chemistry and biochemistry from UCSD, his MBA from SDSU, and his PhD in chemistry from USF. They are going to talk about models to further health in space and on Earth, which is certainly right on topic.
So I think actually Marc is not going to be joining us today, but so you'll just have me, and I'm pleased to be here with you on behalf of the ISS U.S. National Laboratory. To get us started, let us recall that we're celebrating just over 20 years of continuous human presence on our orbital outpost. So that is really exciting. How many young people today have not even been alive while we've not have had presence on the space station? And just briefly, in 2005, Congress designated the US portion of the space station as a national laboratory. Our mission is simple: to enable scientific discovery and technological innovation in space. My company, CASIS, we manage that asset. We manage the ISS National Laboratory. So I wanted to start out by telling you a little bit about the laboratory. Of course, it's 1 million pounds of hardware built in different nations brought together in orbit to make a structure that is about as big as a US football field. It's home to an international crew, and it is also an incredible laboratory to perform research that simply cannot be done on Earth. And of course, you are a very familiar audience, but why space? Why are we doing research in space? Of course, there's 3 unique propositions offered by the space station: sustained microgravity, extreme conditions outside the space station, and a vantage point from which to study the Earth with remote sensing, and of course, looking out into the cosmos as well. And when I'm speaking to potential researchers about doing science on the space station, a lot of people have not really considered that, but I like to tell other scientists that conducting research in microgravity is a lot like looking at your science, whatever your topic is, with an entirely new lens of discovery. So in contrast to NASA's missions to use the space station to benefit further space exploration goals, our mandate at the ISS National Lab is to provide access to the space station to the research community that is interested in benefiting life on Earth. Essentially, bring value back to the US taxpayer. We work with academic researchers, other government agencies, like NIH, NSF, DoD, and industry to carry out our mission. And as you are now very familiar, thanks to our previous two speakers, spaceflight induces a number of physiological changes in our astronauts. I won't go through those because I think you're now very familiar with them. But it is interesting to point out again that this all is happening in otherwise extremely healthy, weird individuals, as you said, Rand. And these changes appear similar to several maladies here on Earth that occur either in our aging population or during injury. So this makes studying astronaut health in space really interesting to people that want to understand onset and progression of disease processes, especially because these changes in spaceflight occur very fast, and also because for the most part, they are reversible upon return to Earth. Which brings me to a topic of regenerative medicine, essentially regrowing, repairing, or replacing cells, tissues, or organs. I live in Houston right now. It's still really hot. There's a bunch of lizards in my yard. If I go outside and I catch one by the tail's going to drop off. Lizard's going to be happy. It's going to grow a new one. How come humans can't do that? Well, aside from the fact that we don't have tails, but why can't we regrow limbs? Well, it's very complex, but part of that answer involves stem cells. Stem cells are unique cells in our body that can develop into specialized other cells like neurons or cardiomyocytes. One day, stem cell-based therapies may be much more widely used to treat a variety of illnesses. Now, we're not quite there yet for a variety of reasons. Some of those limitations include growing enough stem cells for treatment, keeping the cells from differentiating until it is time, safe cell sources, etc. But there is a lot of research going into stem cells right now, even in space. Now, we believe that there is an advantage to culturing and expanding stem cells in space. First, differentiation has been halted or even reversed in microgravity. In other words, stem cells stay STEMmy in space. Second, microgravity improves stem cell growth and expansion. Third, we can use stem cells in microgravity for disease modeling or manufacturing of tissues and possibly organs. So stem cells enable us to model diseases that are specific to a patient, the precision medicine that Dr. Fogarty mentioned. Imagine if you are an astronaut and you're about to go to Mars, wouldn't it be interesting to send some of your cells, some of your stem cells to space first to get an idea of what kind of countermeasures your unique physiology is going to need? Stem cells also enable us to do some biomanufacturing of tissues and organs. We may be able to take a patient's own blood cells, revert them back into stem cells, then reprogram them to become any other cell in the body. And because they are uniquely from a unique patient, there's no concern for immune rejection if you have printed an organ or tissue. Now, we've tried to use 3D printers here on Earth with moderate success, but we're limited to about 1 centimeter of thickness because the weight of the tissue collapses on nascent blood vessels, and it's really challenging to get thick tissues. You need to have a scaffolding, et cetera. But in microgravity, we don't need a scaffolding. There's time for angiogenesis and maturation of tissues. And just imagine, there's over 100,000 people just in the US that are waiting for an organ transplant. Imagine being able to bring that number down. Now, we are a very long way from custom printing organs in space, so I don't want to oversell that. But perhaps we're not too far away for providing organs for drug development and testing. Another topic that is relevant to stem cells and models in microgravity are tissue chips or microphysiological systems. These are engineered silicon or some other biocompatible plastic device that can support living human tissues and cells in a 3D matrix with media and replicated mechanical forces that the cells would normally experience in the human body, such as lung inflation, deflation, and pulsatile blood flow. Because of all those things, because of the 3D nature, because of the mechanical forces, tissue chips recapitulate or mimic complex human physiological responses better than standard 2D cell culture in a flask. And thus, we like to say that tissue chips give cells a home away from home so that they behave more like they do in the human body. And I like to use the analogy that ISS gives astronauts a home away from home as well. And since you may be using a specific patient's cells in a tissue chip, the tissue chip will have the same disease, if you will, that the human has. So if you want to study a heart arrhythmia, for example, well, a tissue chip with cells from that patient, if they're cardiovascular cardiomyocytes, will also have a heart arrhythmia. So tissue chips can really model specific diseases. And in addition, we can connect tissue chips. So you can have a brain chip connected to a liver chip, connected to a heart chip. You can test the responses of several organs at the same time. And an eventual goal is to have an entire human body on a chip or a series of chips. Real briefly, why is the NIH so interested in tissue chips? They've put a lot of money into this. Well, in the United States, just to develop a new pharmaceutical agent, a new drug, it takes 10 years or so and maybe $2 billion for an idea to medicine cabinet. And many of those, about 1 in 5,000, make it from preclinical testing all the way to market. So there's a huge failure rate. Now, that's good. We don't want to short circuit anything about our testing process. The FDA in the United States protects us from drugs that are either toxic or inefficient or ineffective in humans. But we'd like to be able to speed or make that process more efficient. So imagine being able to hook up a few 100,000 human tissue chips as opposed to clinical testing in humans. Maybe we'll make this a little more efficient, more relevant, and more ethical. Maybe we can even eliminate animal testing. Now, we're not there yet. Tissue chips are only about 15, maybe 20 years old, but this is really cutting edge bioengineering. And the NIH is pushing the technology to make this, validate the science, develop some standards, and make the tissue chips more turnkey in a laboratory. And that is where tissue chips in space comes in. If you want to study a disease like bone loss on Earth in a tissue chip system, well, it can take years or at least many months for a disease process to develop. Well, we know that astronauts begin to lose bone in just a few days on orbit. Disease processes, of course, are accelerated in microgravity. So maybe we can use tissue chips to study that disease onset and be able to address these issues quicker. And as a bonus, in order to fly one of these experiments to the International Space Station, we have to kind of modify and fit an entire laboratory into something about the size of a shoebox that is going to be operated potentially by a fighter pilot. So a lot of work is done toward turnkey and automation. And real briefly, a lot of this funding is coming from the NIH Center, the National Center for Advancing Translational Sciences, NCATS, and also NIBIB, National Institute of Biomedical Imaging and Bioengineering. In partnership with NASA and the ISS National Lab, we've got nine different projects that are each flying twice to the space station. So this is really exciting work. And many of these have already had their first flight and are working on their second flight. So I think look forward to some publications coming out soon, maybe in the next year or so. There's another model that I want to briefly mention that can be used in microgravity, and these are organoids. So it's kind of between just 3D culture. Well, they are 3D cultures, kind of between 2D cultures and tissue chips. These are self-organizing 3-dimensional tissue cultures, so that the cells get the interaction, the cell-cell interaction, cell-cell communication. They can mimic simplified organs, and these are also becoming more and more commonly used in disease and development testing. Real quickly, I wanted to mention just a couple of other science research results that may interest you. The first I'm going to talk about has to do with protein crystal growth. I'm sure a lot of you have heard about that. There's a couple 100,000 different proteins in the human body. And if you understand their structure like really well at the atomic level, you can design a drug to mimic or enhance or inhibit that protein function. Trick is, it's a little challenging in many cases to grow protein crystals on Earth well enough to understand their atomic structure. Basically, gravity is a distortion factor, but you can grow the crystal in microgravity. It can grow more perfectly, more largely, or more uniformly, and then you can really understand the structure. So Keytruda, this is a drug out of Merck pharmaceutical company, is a very commonly prescribed cancer drug. And the kind of issue with it is, as a patient, you have to go into the clinic every couple of weeks, get an IV infusion, stay there for a few hours. There's some inconvenience there and some practical limitations. So Merck Pharmaceuticals sought to improve the drug delivery method and patient quality of life. So their microgravity experiment that was done during Expedition 50 sought to improve those things. And they were able to show that they made a more uniform suspension of very small crystals. And now they're working to mimic their results back on Earth. So this is not something that has to always be done in space, but essentially make this drug formulation easier to use and improve patient quality of life. Here's another model, cancer therapy. This is a drug that is being developed by a small company, a small biotech firm called AngiX. They have an idea to use cultured endothelial cells in microgravity as a model, an in vitro model for resting normal endothelium, which is a very quiescent, reduced state of cell growth in resting endothelium. We don't have a great model on Earth, so they decided to test their cancer therapy, their chemotherapy agent in microgravity, and their results were very promising. And they used very high doses of the cancer drug that on Earth was killing their stems, their cell culture, but on Earth, in space, this drug seemed to work very well and did not kill the cells. So again, using microgravity as a model system for more physiologically relevant tissues, cultures, models, etc. One more interesting science result, kind of in the biomanufacturing realm, comes from another small biotech firm called Lambda Vision. So they make a protein-based retinal implant, a prosthetic, to restore vision to millions of people who have some kind of retinal degenerative disease like retinitis pigmentosa or age-related macular degeneration. So they make these retinal implants on the ground. They make it on Earth. It consists of multiple layers, kind of a layer-by-layer approach of a polymer and an ion permeable scaffold. But gravity, again, interferes with both the homogeneity and that uniformity by encouraging sedimentation and gradients of the solutions. So they tried this in microgravity and had some really good luck demonstrating that they're able to make better, more stable and more uniform retinal implants. And so now they have received a large grant from NASA's commercialization efforts to continue this work. One more model that I want to mention really quickly is a rodent research capability on the space station. We can bring up to 40 mice at a time. Sounds like kind of a small number for if you're working in a major pharmaceutical company. However, it's better than nothing. And we're able to do some pretty advanced interventions with these mice. And we can do DEXA scans on orbit. We can do grip strength measurements, so we can measure the same physiological changes that are happening in astronauts. But the mice, the benefit there is they don't have exercise countermeasures. So if you really want to study muscle wasting in a full physiological model or bone loss, for example, you can do that with rodents. Lastly, kind of hopefully to try and catch us up on time a little bit, we do have a lot of ISS National Lab research announcements. There's a web page at the bottom of the slide there where we're going to be posting all of our solicitations. So if you're interested in doing some research in space, this is a place for you to keep your eye on. And also the link up above, we just published it. This is in preprints, a manuscript based on a symposium we held all about opportunities for biomanufacturing in low Earth orbit. A lot of those models I talked about are all included in this position paper and may be of interest to you. And that is all for me.
Thank you, Liz. I do appreciate you're keeping us on time, or at least not getting further behind. And I should say, please keep an eye on chat, because I'll be giving people time warnings there, as you're talking. That may help, how much, where you are in terms of schedule.